Okay, hi everyone. I'm Rosie Kaplan, and I'm very excited to be here. I want to thank all of you, the speakers and the audience, especially from all the doctors and patients out there that really appreciate the work that you're doing and the people that are thinking about it and asking questions too. I feel badly saying this. I'm not speaking for all doctors. I'm only speaking for myself when I say we're pretty low tech compared to the really high tech cool talks we're talking about. And I want to kind of take you through the world that I have as a pediatric oncologist a little bit, and how I span the clinic and the lab work, and why your work is so important to all of the things I do. And I apologize for the, I guess, low tech aspects of some of the parts of my talk. Okay, so we got very interested in spatially understanding the microenvironment because my mentor was very visual pathologist by training before he became an oncologist. So I got used to looking at pictures. And we love this one. This was one of my students got this on Valentine's Day. And of those of you who like to look at images can show it's a heart, which is the imaging that we did of the pre-metastatic niche. And I was super excited because one of the companies, vendors had a poster of the pre-metastatic niche. And considering I made up the word, I was pretty excited to see it in a poster. Okay. Can I move? Okay. So the big question that drove me at the beginning of my career and still drives me today is, what are the key mediators of metastasis? And metastasis, for the most part, I'm speaking about solid tumor cancers, and I'm pretty cancer agnostic, but I do take care of patients with sarcoma, so I use that. And this is a bone cancer in a 14-year-old boy who was playing soccer and developed pain, and they got an X-ray and they found this. And what I mean by gross is that it clearly was visible on the X-ray and also easier to be seen on more sophisticated imaging. And then the family was devastated, obviously, by the news, but happy because it appeared to be localized. And the majority of patients that get diagnosed with any solid tumor cancer, it is almost most invariably likely to be localized at diagnosis. That's anywhere from 70 to 90% of patients, depending on the cancer type. And we do chest x-rays because the most common site of metastasis in sarcomas is the lung, which is also a common site for many epithelial adult cancers, common ones like breast, and even lung is metastatic to the lung sometimes. So we do imaging, in this case, the CT. But unfortunately, most of the patients that do progress and die of cancer is from metastasis. They get spread to the lungs and other sites of the body. And I think there's really no site of the body that's off limits to metastasis. I think we don't tend to do imaging once patients get very advanced disease, so we don't get to see those very interesting places. But the main questions that I had as a young investigator was to understand this process on the cellular level. What is happening from the time that patient was localized to metastatic? And we found that there were some key events. And we called these key events pre-metastatic niche. And when I refer to that, it doesn't mean that there weren't disseminated tumor cells. But clinically speaking, that patient did not have metastasis when we diagnosed that patient. There was no evidence on a CT scan of a nodule or a lump or some evidence of metastasis, but clearly there were events going on. And I hope this shapes the rest of my talk that's really about these events. And to understand what we call pre-metastatic niche doesn't mean that there weren't disseminated tumor cells. It means more that there wasn't clinical evidence of metastasis. And this process, now that we understand of these events that really focus on stromal plasticity and ECM remodeling, myeloid infiltration, and immune suppression, many themes we've heard throughout the day in the talks were occurring early on in the metastatic process, and probably also occur very early on in the tumor formation process. But in our cases, most of our patients don't tell us before they get cancer, so we can't really track that. And we don't have good things like epithelial cancers where you can get DCIS or something like that. For a sarcoma, there's no such understanding of that. Okay. So that's our main goal. And I think we always wanted to come back to the heart of it. Even though I don't have exciting spatial data to show you, I will say that we've been always focused on the architecture. And I hope this is an area of opportunity to collaborate with all of you more on the architecture of that microenvironment. Okay, so we know it's a stepwise event. And I have to say, if I distill out all the years of my lab now at NCI, first at Memorial and then at NCI, I would say that there are basically two processes. And mathematically, I'm sure a lot of you could model this, and I'm kind of excited about that idea. But if I was to distill it down to two processes, immune suppression is essential. You need that for the metastatic process. I don't care anything about the genetics of a cancer or its plasticity or EMT or anything else. And I promise you, I'm not tumor agnostic in the sense that I care about cancer and I care about cancer cells, and people should be doing the genomics, but you have to have your host has to be immune suppressed to have this happen. And you need fibrosis. And it's microfibrosis and it's microimmune suppression. You're not overall immunocompromised and you're not overall having fibrotic disease, but you need to have those events. And that allows for metabolic reprogramming and plasticity. And we always think of plasticity in a very distinct way. In the cancer field, many people refer to plasticity as the tumor cell plasticity. But what I'm talking about is the plasticity of our microenvironmental cells, which are the cells that make up your cancer, actually. But those cells are used to being plastic, and that plasticity is essential for the plasticity of the tumor cells. Okay, so my whole talk's going to be about these two events and basically explain what we think the pre-metastatic niche is, a specialized microenvironment that happens at distant sites before clinically detectable metastasis, and it involves multiple cell compartments. And as she said, don't add all the cells in together, but anyway, the body added all the cells in together, so it's really hard to tease apart, and I get stuck with this. And we've been focused on myeloid cells, one, because they're found in virtually every solid tumor. And I ask this in every one of my talks. If anyone ever finds a cancer that doesn't have a fibroblast or a mesenchymal cell type or a myeloid cell in that cancer, I need to study it immediately. Come find me, call me, anything. I don't care if it's 2 in the morning. I want to see it. And I think that they clearly play important roles and diverse roles depending on the tumor and the microenvironment, but they're invariably there. We either have myeloid cells and mesenchymal cells there, and they contribute both to pro-tumor and anti-tumor functions. Myeloid cells are not necessarily as chic as T cells right now because people love T cells, have the real functional effector power, but the nuance that myeloid cells has is they can really read the room well and they can act accordingly. And that's, I think, a really special gift of myeloid cells. We know that that the immune system is capable of recognizing tumor cells and damaged signals. Mostly we think of them as T cells that have a TCR that can recognize it very specifically. But myeloid cells can antigen present, and that's essential for T cell activation. And alternatively, they can really dampen down the signals. So I think they're really shaping this process. And essentially, a lot of work went into showing that these cells, myeloid cells that come into the tumor, a lot of them come from the bone marrow. They come from the bone marrow during hematopoiesis. They get out into the bloodstream. We can measure them in circulation in mice and in people, and then they form these clusters. So when we do single cell, which I've done, and I'll show you data of that, we digest the whole tissue. But when we look spatially, we knew that it wasn't just that there were a lot of myeloid cells, but that they were clustered together, and that clustering is important. And I still don't understand the difference of these green cells and their clustering and their behavior versus myeloid cells that are distant from that slightly or greatly, depending on where they are and how their behavior is different. And it's an easy answered question for a lot of you, but not for me yet. So I think that's big on our radar. And then when we look at this, at least by single cell, these are sarcoma models that are put in the orthotopic site and metastasize to the lungs. When we look in the pre-metastatic lung, we see that the biggest pathway upregulation is downregulation of adaptive immunity. So myeloid cells suppressing T cell activation and cytotoxicity, and inhibition of antigen presentation. And we know that they're myeloid mediated because most of these pathways are coming from the myeloid compartment by single cell. And much of this work was done by Sabina Kachanowska to look at these changes by flow cytometry, which showed this expansion of myeloid cells and a contraction of T cells, in particular CD4 T cells, as well as dendritic cells. But now we've been fortunate to have a good collaborative team computationally with Sridhar Hanahali and Vishaka Gopalan to help us understand it in patients. Because I'm a doctor, I want to take the pre-metastatic niche from my mouse models into patients. And we find that when we take patients with, in this case, adrenal cortical carcinoma, which is a very rare epithelial cancer that is a neuroendocrine type tumor, and it metastasizes very heavily to multiple sites, including lung and liver. I thought, well, maybe we can look at this metastatic tissue and look at the organ where there's metastasis, but not in the tissue that does not have evidence of cancer, to understand the biology. And we hypothesized that this metastasis-free tissue in a metastasis-bearing organ could be conditioned by the cancer and could have features of the pre-metastatic niche that we found in the mouse models. And what we can say, I think, and pretty convincingly, is that the metastasis-free tissue, architecturally and transcriptomically, is not the same as cancer-free healthy tissue. And so that your microenvironment is changing early on in the metastatic process, and that most of these patients that have metastasis will go on to metastasize. So even though they're advanced metastasis, they also have new ongoing metastasis. And so that's the biology that we thought. And what we found, sorry, this has gone crazy, sorry. And what we found together was that there was classical monocytes, similar to the recruited bone marrow-derived myeloid cells we found in murine models, were present in these tumor metastasis-free tissues from patients, this cancer-conditioned microenvironment we call CCME, and that they were dampening down adaptive immune responses, and that they were very predictive of a poor outcome. And by TCGA, looking at those outcomes, tumors that had this signature did very poorly. So if you had this classical monocyte immune suppressive signature, it was basically worse than almost any genomic feature you can think of. And so at least it reassured me that these cells are important for going after that. I don't know if I can convince you here on the screen if you can see, but we've started to do some IHC, simple, single and then multiplex IHC to understand what these tissues look like. And I think we can say that the cancer condition metastasis-free microenvironment does clearly not have evidence of metastasis, but it does look architecturally different than the metastasis tissue. And there are computationally, we can see that there are clear differences in the transcriptomics of these tissues. So I think we're starting off on the right foot that way. And we compared, we've subsequently compared our murine models where we've developed a gene signature of this immune suppression and of the pre-metastatic niche, and find that signature in the human tissues as well, and heavily immune suppressive in the key features. And so we take this as a big hypothesis. I'm a doctor. I want to treat patients. How can we reverse this immune suppression? And I think a lot of people had strategies, but this is now kind of early on in the process. About 5 years ago, we were like, okay, can we do this? And so I thought, well, myeloid cells are great at getting into tumors. They're really good at reprogramming the immune response. Why don't we engineer them to do what we want to do? And so we made them deliver factors, and we've been studying them. We call them GEMMs, genetically engineered myeloid cells. And we found that IL-12 was the most downregulated cytokine in our pre-metastatic niche, and so we focused on IL-12. It's also important to note that a lot of people like therapeutically cell therapies, they want to give cytokines to the cell therapies like CAR T cells or TCR T cells. But naturally, your myeloid cells make IL-12, and the T cells express the receptor for it. So it seemed to me make more sense to have the myeloid cells make them. And when we give these genetically engineered myeloid cells, I'll call them IL-12 GEMMs, into mice, we see that we can restore their anti-tumor T cell efficacy very effectively, both with T cell increases and K cell cytotoxicity and CD4 help, which is critical to the process. When we give these IL-12 GEMMs with cyclophosphamide fludarabine conditioning, which is the typical chemotherapy conditioning we do with CAR T cells, we can cure these very aggressive, large metastatic tumors. And before this time, I had never cured a mouse. And because we had these mice cured for so long, we decided to re-challenge them, and they remained immune to the tumors and kept alive even after that. So we are very excited about this work, and we are doing more ongoing work now to show the endogenous memory formation and how this works. We also have done more recent work looking at osteosarcoma. That other model was a rhabdomyosarcoma, different sarcomas, but same kind of biology. And now we have a work now in breast cancer that looks similar in these very T cell poor and myeloid-rich environments like this, there's really a predominance of myeloid cells. We can see that a group of them are highly immune suppressive. And when we're looking at cyclophosphamide and fludarabine in particular, we find that cyclophosphamide and fludarabine is critical to reprogramming that myeloid compartment by getting rid of these immune suppressive cells and regenerating the myeloid cells coming in to be more sensitive to immunity by being exposed to the IL-12 GEMI. So that combination we think is highly potent in eliminating the MHC2 negative monocytes that are immune suppressive and activating these MHC2 positive macrophages. So you need the myeloid cells there. You don't want to get rid of them, as a lot of people think. You get rid of them, but you need them to come back, as we heard this morning too, essential for them to come back and present antigen. And so we see that, and then we get effective anti-tumor CD8 T cell responses in this process. So we're dying to go to the bedside with this. We've prepared, we're ready to go to the FDA. We're just waiting for NCI to help us move this along because the administration decided to have a lot of people depart at the time that are functional to this process, but we're hoping to get there very soon. And so we have a single arm phase 1 trial of IL-12 GEMIs, and we spent a lot of time. All the studies I showed you were in mouse and using mouse GEMIs. Now we have human GEMIs, and we're hoping to give it to patients. And the exciting part is we want to do a lot of bedside, back to bench correlative studies to understand this biology by doing pre and post biopsies after treatment with the cell therapy. And those biopsies, I hope, will have H&Es, and we'll have tissue, and we'll be able to do spatial transcriptomics, and you'll tell me why it's not working, because I love your mathematical models, but my patients never seem to follow math rules, and I don't know why. I tell them to read the book, they just don't. So it would be really good to know if there's some nuances or some crazy things that this responses is different than what we've modeled, and be able to model that better. Okay, if I don't know how much time I have. Do I have the time? Okay. I'm going to talk for 2 more minutes of this, and then I'll stop because I won't have enough time. But the other thing that I talked about was really important in the pre-metastatic niche, and that was you need stromal cells there, you need stromal cell plasticity, and you need extracellular matrix. Those things all together are essential, and they're connected. So it's not like they're separate events, they're all events that are connected to each other. And the early days, we saw that you needed this upregulation of fibronectin. And I have to tell you, when I first discussed this idea that fibronectin could be upregulated, the only person who believed me was Richard Heinz, and God bless him. I just rest his soul because I just heard about his passing, but he was an incredible mentor, and he really believed in me because fibronectin is ubiquitously expressed throughout the lung and throughout the body. And when I raised the idea that there could be focal pockets increased of fibronectin, every time I did this, nobody bought that. But, it was before spatial transcriptomics, it was before single cell, and it was hard to prove. But by IHC, we could definitely show it. And I think thankfully to him and his support, we were able to publish that and still been working on it. But then we really wanted to say, what were the key effectors of this? And so I spent a good portion, 10 years or so on the effectors. And I'm not going to get into that now, but Mira Murgai was my postdoc and now has her own lab at NCI. She's interested in pericytes, and now we're interested in Schwann cells. So if you want to know about the effectors and their function and how the ECM are modeled, I promise next time I'll talk about that. But the one cool thing that we were interested in is what else besides fibronectin was involved in this ECM? Were there other components of the ECM? Because collagen was there, but it's collagen 1, which is the one that everybody loves to look at, was the least changed. And everybody asked me about collagen, and I would look, and I was like, okay, I need to look at all these other fibrillar collagens. And we would try, and transcriptomically you can find them, and it's interesting, but they didn't change as much. And what we found changed the most was hyaluronic acid. And hyaluronic acid is not really part of the traditional ECM pathways that you look at when you come, so you don't find it. But when you look here, and I don't know if you can see, but it looks a heck of a lot like where the myeloid cells were going when I would say they were clustering to form this pre-metastatic niche. And this is a similar model. So we said, okay, hyaluronic acid is really important, and they're really important for myeloid cells. And so Alice Brown, my graduate student, now postdoc, stayed, and I said, why don't we genetically engineer the mesenchymal cells to alter HA to understand its function, both its functional and see therapeutically. So we make what we call gemesis in order to understand the role of hyaluronic acid and extracellular matrix remodeling in general in this process. We thought it could locally break it down. They're the cells that make it, could improve fibrotic tissues, may improve drug delivery. And so we did this. And so when we give our mouse models hyaluronidase expressing gemesses, so we genetically engineer the mesenchymal cells to make an enzyme that breaks down hyaluronic acid called hyaluronidase. We use SPAM1 because it's very pH insensitive. We find huge changes in collagen and HIF. And basically, you can see here real acute changes in hypoxia that happen very fast. In fact, too fast for vascularization, more likely a normalization of the vasculature or a metabolic process. And then when we deliver it, because we want to understand if it had any therapeutic potential, it seems to stave off tumor growth when you add them like that. And we thought, okay, well, we're going to give one or we're going to give multiple. This was one dose. We had some effect. So then we said, okay, let's do multiple doses and let's give it with chemotherapy. And you can see when we give it with standard chemotherapy, which is doxorubicin cisplat for this particular tumor, and these genetically engineered mesenchymal cells, we see really remarkable anti-tumor effects. And so we're pretty excited about exploring this further. And therapeutically, we know that we can also see huge changes in the immune compartment. And so there's lots of crosstalk between the myeloid cells and the ECM remodeling and CAF associated events. So we're hoping to look more into that. But in conclusion, I'll say, I think there's a lot of power and potential for modulating the tumor microenvironment as a therapeutic avenue, in addition to the strategies that we focus on the tumor. These disease microenvironments have abundant myeloid cells, activated stromal cells, and matrix remodeling that work together. Understanding these dysregulations are going to be critical to getting these therapies. We're really hoping to evaluate the safety and feasibility of human IL-12 GEMIs in patients very soon. And hopefully we can just explore the spatial architecture more after the therapeutic effects to see if that explains the benefit or lack of benefit in patients, because I'm very realistic to that, to failed clinical trials. And then I think ECM remodeling is going to be the next frontier for us therapeutically to try to understand. We took a kind of different approach without, we didn't want to kill off CAFs. We didn't want to focus on one particular ECM component that was, critical to the collagens per se. But we think that this kind of delicate rebalancing may be an effective approach. And then I think because TME is such a collaborative process, we really need to understand how to combine these therapies effectively and how we would give each one and what the impact would be. And I really want to thank all the people that did all this work. Sabina Kaczynowska did most of the GEMI work, along with Jim Kronk, who's now has his own lab, and Alice Brown did all the GEMESI work. And of course, the great collaborators in pediatric oncology branch, they're the reason I can stand this current environment the way that I do. I have my own little microenvironment that's okay, despite the larger environment, and definitely lots of collaborators, and in particular, Center for Cellular Engineering that helped us to really make the human cells for clinical use, which was a big arduous task. Thanks so much for your attention.
Questions? Here in the corner. Oh, I'll be back. I'll be back up. I promise. Over there first or here? Okay. It's crazy. I'm from NCI as well, and it's really exciting to hear the overlap. So we have spatial transcriptomics data, Visium HD data from Kaposi sarcoma patients before, during, and after IL-12 treatment. So we should talk. We would love to explore. About half of the patients are responding to therapy, and we would love to hear your hypotheses, and we can dig into the data and see what's valid.
Thank you. That would be great. I have to come to UMD to get NCI collaborators.
It's awesome to hear that roll out. And, I was curious with your talk with the hydrochloric acid link, and given that inhibiting that has had such a checkered history in promoting metastasis, I'm really curious to get your sense of that.
Yeah. I think this is also true for IL-12. There's lots of people that are going and trying to deliver IL-12, whether it's through tagging it to, to a molecule that keeps it in a depot form, or putting it in a T cell. And I think there's all approaches have valid reasons, but I'll kind of give you the 2 cents of why we did what we did. Myeloid cells make it, and they make it at a very certain level. And I think it's not only important that you make it, but that the cell that makes it is communicating with the other cells that it normally communicates with. And that interaction, myeloid T cell interaction of making that, I think is critical to the biology. And I think we miss it when we have another type of environment trying to simulate that. I mean, math is math, so I don't want to say IL-12 wouldn't have its effect. Okay. Okay. Yeah. Sorry. Yes. So SPAM1 was pegylated and used in pancreatic cancer. And the main issue that I understood, and I may be wrong about this, but the dose that they were able to give that the translational researcher who did the original work never felt like they could ever get to the dose that he was able to get in his preclinical models because of the toxicity of the adjuvant in that pegylated hyaluronidase form. So that can be a problem both of delivery and of, alternative things. So I think that checkered that history, at least for pancreatic cancer. For the data on the patients that did respond, he did see changes in hyaluronic acid, and that was associated with response. So I was actually bolstered by that. I think in this case, we're talking specifically about levels and low molecular weight versus high molecular weight and what it means. But the cells that are normally doing the remodeling may have a totally different effect than trying to deliver a drug and get it to where it needs to be and get it at the doses you need. So I think cell therapy has its own innate biology that's going to help us there. So I sort of taking that bent to it. But I think at this point with the Gemesi's, we need to do more preclinical work to really get at that. And that's where I would love to collaborate with people that can help us. I would love to just give cells in a dish to see its, in an organoid model, let's say, and computational workout. What is it actually doing? How is it doing it? Because you could do a million in vitro and in vivo experiments and not get to that answer the same speed.
Question?
Hi, that was a great talk. I was just wondering if you guys have at all worked with chondrosarcoma biopsies or like skull-based chondrosarcomas at all in your work with the GEMCs?
So, sorry, which kind of sarcoma did you ask?
Chondro? It's a
Chondrosarcoma, yeah.
rare yeah.
Yes. I definitely have had chondrosarcoma patients. In particular, the biopsies in general are dealing with the tissue. Is that what you're speaking about? It's being a challenge. Yeah, I think it is definitely a challenge. I'm not sure it's more of a challenge than osteosarcoma, which is a huge challenge. But I actually would refer you to Troy McEachran, my colleague and friend, who has actually done incredible spatial Visium HD spatial transcriptomics on osteosarcoma successfully. And we have chondrosarcomas in that cohort as well. And he would definitely be the person, and I'm happy to connect you. But we have done it successfully. And I think it's quite doable. And I think it's ripe for that because clinically, when I said low tech, like we are so low tech in the space, like with sarcomas, because they're so difficult in terms of that, like digestion and all that, that I think it's prevented us from really making the progress we need. But we are starting, and I feel very hopeful that we will prevail with that.